Ions are atoms that have either lost an electron or gained one and hence have either a positive or negative charge. If the ion has lost an electron, it has a positive charge. And if it has gained an electron, it has a negative charge. Why? Because electrons are negatively charged. Okay, just a quick recap from GCSE. Water can act as a solvent, which means it can dissolve the solutes. Okay, ions can act as a solute. They can be dissolved by water when the force of attraction between the ions and the water molecules is stronger than the force of attraction between ions. Okay, so we're gonna look at a diagram of that. It's gonna help you understand. Just another definition here. A solution is the homogeneous or even mixture formed when the solute is dissolved in the solvent, okay, or by the solvent. Now, remember that water has a slightly negatively charged oxygen. This links to a 1.1, the first topic you've ever done, okay? And because it's negatively charged, or at least slightly negatively charged, it's attracted to the positive ions because we know positive attracts negative and vice versa, and that's electrostatic attraction. So because it has this slightly negatively charged oxygen, it can attract the positive ions. And because its hydrogens are slightly positively charged, it can attract the negative ions. Okay. Now, in the case of covalent compounds, such as glucose, they can also dissolve in water, but this time it's not going to be via electrostatic attraction. This time it's going to be because of intermolecular interactions. Okay, and remember that water is dipolar. That's why it has the slightly positive or negative charges. Now, we're going to go back to why the ions can dissolve in water and when they dissolve with this diagram here. Now, this salt crystal contains Na plus sodium and Cl minus chloride. Now, there's a positive ion and a negative ion, and this causes electrostatic attraction, forming this salt crystal. Now, when in contact with water molecules, we know that water molecules have a slightly negatively charged oxygen and a slightly positively charged two hydrogens, right? Now, the oxygen, hence, is going to be attracted to the sodium, and the hydrogens are going to be attracted to the chloride, okay, because of the opposite charges. And when this force between these water particles is stronger than the force holding this salt crystal together, it's going to eventually dissolve it. Okay, it's going to form the solution. All right. You should know from other topics that osmosis is the net movement of water molecules from a region of high water potential to a region of low water potential across a partially permeable membrane. So imagine that this line in the middle right here, this black line separating these two sides, is a partially permeable membrane, because we're going to look at an example of osmosis. Now, water always flows from hypotonic solutions. Okay, imagine this left side is hypotonic to hypertonic solutions until they become something called isotonic. Okay, this is going to make sense in a second. So remember, water flows from the high water potential, there's more water on this side, to the low water potential. This is osmosis. So hypertonic solutions have high water potential and low solute concentration. Hypertonic solutions have the opposite. Okay, they have low water potential and high solute concentration. So remember, the water goes from high water potential to low water potential via osmosis. Now, I mentioned mentioned the term isotonic before. And this is when there's an even amount of solute concentrations, okay? So what does this mean? Well, first understand that solute concentration is how many solutes compared to the water volume. So we saw before that this left side had a lot more water and it only had 3 of these, right? It only had 3 solutes. So it had a lot of water and little solutes. While this one had little water and a lot of solutes. So if we give this side the water, they're going to both have an equal solute concentration, more or less, okay? Moving on. Oh, forgot to mention. While they are now equal, there's still going to be movement of water, right? There's going to be movement of water back and forth. Maybe it goes from here to here. But at the end of the day, they're going to be in dynamic equilibrium. So they're always going to be moving, but it's always going to be equal. So the dynamic is for it's moving, and the equilibrium is for it's even. Okay, so isotonic doesn't mean it stops moving. Isotonic just means that it keeps moving, but it's in equilibrium. What happens if you place an animal or a plant cell in these 3 different solutions, these environments? Well, starting with isotonic, because there's dynamic equilibrium, it's balanced, the cells, the animal cells, are going to be in a normal state because it's isotonic. Okay, there's nothing wrong, it's just there's balance. And the plant cell will just stay in a state called flaccid. Now, what about in a hypertonic solution where there's low water potential, but there's a lot of solute concentration? The cell, the animal cell, is going to lose water, it's going to shrink. Why? Well, this is because in a hypertonic solution, remember, there's low water potential. This means that the animal cell probably has a lot more water inside of it, and we know that water moves from high water potential to low water potential via osmosis. That's why the animal cell loses water. Will the plant cell lose water? No. The plant cell undergoes a process called plasmolysis. We know plants are very adapted to water, okay? So they have these adaptations such as plasmolysis in order to prevent the loss of water and, shrinking or cranating. That's a nice word to remember, cranated, unlike animal cells, okay? Now, what if these two are placed in a hypotonic solution where there's a high water potential and low solute concentration? Well, as you saw right here, the animal cell actually swelled, it got bigger, and it burst, okay? It's got a hole right here. It underwent lysis, which is like a breakdown, okay? And this is because the hypotonic solution it was in had a lot more water than what was inside the cell, and so it went from the high water potential of the hypotonic solution inside the animal cell that had a low water potential compared to the hypotonic solution, if that makes sense, okay? And this means that the animal cell was having too much water at once, so it like inflated like a balloon and just burst. Now, what about the plant cell? Okay, this change of color just means it's undergoing turgor pressure, okay? Again, the plant cell this time is also adapted to not burst. Plants are adapted for this, okay? So it undergoes turgor pressure, which is exerted by the cytoplasm against the cell wall. That's an adaptation that they have. And hence they become turgid. They've gained water and they become turgid. We know that flaccid is when they don't they're like they don't have like much water, right? Uh, so this is what it means. Plant cells and animal cells are eukaryotes. But what about prokaryotes? What adaptations do they have if they're placed in a hypotonic or hypertonic solution? Well, these unicellular organisms have something called contractile vacuoles. So imagine a bacteria in the water. It's going to have this structure, okay, to survive. There's not much detail you need to know. Just remember contractile vacuoles. In medical centers, we can actually use isotonic solutions because as we saw before, the animal cell is just in a normal state when placed in one of these solutions. It doesn't become cranated or small in the hypertonic solution, and it doesn't swell or burst or undergo lysis like it would in a hypotonic solution, right? And so these are ideal to firstly conserve organs, which are made of animal cells, right? Uh, and also for something called intravenous injections, okay? These allow rapid and direct absorption of medications, fluids or nutrients, etc. into the circulatory system, and this is what you might have seen at the hospitals right here is literally an isotonic solution. And intravenous basically means that it's injected directly into the veins.